So thank you for that kind introduction. We're both pleased to hear. I'm joined by my colleague Jason Flannick, who co-leads this activity with me. He is assistant professor of pediatrics at Children's Hospital Boston and also an associate member at the Broad Institute, and leads the scientific and technical components of the portal. So he'll be here to address questions as we go through. As it was said, if you want to pop any questions you have as we go in the chat line, I planned a demo of the site, but not a live one, because you know how live demos often go. They tend to be slow. I did it as screenshots so that we can go through a little bit more material. So if anyone has any questions, feel free to pop it, but I think I left some time for questions at the end in the talk. So what I want to share with you today is the AMP Type 2 Diabetes Knowledge Portal, which is an open access resource for providing data and tools to promote our understanding of type 2 diabetes and its complications. And if you'd like to take a look while you're on, the URL is listed below. It's open access and anyone can access it, but I'll be walking you through it, so we can wait till then if you want to. So before I begin with the actual demo, I want to walk a little bit through their motivation, talk a little bit about how it's built, some of the foundation of it, because I think that really helps as we go through some of the tools and modules for you to understand what the underlying data and architecture is to the resource. And finally, we'll talk a little bit about what's come and some related resources that I think will be of interest to you. So first, kind of where we come at this perspective, our motivation for this is rooted in the foundation of human genetics, because I think we feel very strongly that in order to understand disease biology, or develop better therapies or prevention or prediction mechanisms, it requires experiments, and the best experiments in our opinion is human genetics, because these are experiments of nature, because they're natural perturbations or variants that occur in our genome that we can systematically test. We can systematically test these through genome-wide association studies and also sequencing studies. And by testing these perturbations, we can learn direction of effect, magnitude, significance that points us to variants or genes that may be implicated in the disease etiology. So that's really the foundation. That's where the resource kind of got its foundation and both its roots. And we are definitely not limited in our current, in 2020, by the amount of genetic data out there. You know, routine genome-wide association studies now are numbering in the millions of samples. There are national and international efforts to generate large-scale genetic data sets linked to medical health records with rich phenotype data. You've probably heard of many of these: Million Veterans Project, UK Biobank, all of us, FinGen. They're both international and national, and they are mounting up large efforts to bring genomics into healthcare. So we're not limited by our data. We're also not limited by motivated research communities. The type 2 diabetes genetics community, which we've been involved in for many years, close to 20 years for me, has worked for many years, starting with linkage studies back in the early 2000s, all the way up to large-scale sequencing studies to generate large-scale data for type 2 diabetes, for sequencing and genotyping, and work together to understand what loci might be implicated in type 2 diabetes. And there's been some pretty great success from my perspective. Over 403 loci have been associated with type 2 diabetes to date. This has been in samples, as I mentioned, about 1 million samples is the largest data set currently available. What this means is that we've discovered a lot. We have no idea what a lot of these loci do. And the story is similar for a lot of complex diseases. And I put this in there because I wanted to give you a sense of like what we're up against. It's a major challenge. So this light up chromosome plot shows you about 100,000 associations across the genome for complex diseases. And I sort of highlighted some of the diseases that we at the Broad Institute are deeply involved in, and then highlighted even more some that you may be particularly interested in from a diabetes perspective, because they're involved in diabetes and related traits. So for coronary artery disease, you have 166 loci, for example. For atrial fibrillation alone, 100 loci have been uncovered. But in many cases, what these genome-wide association studies do is give you a loci, a region of the genome. Sometimes this region is huge. It points to common variations. It doesn't tell us what is the variant or what is the gene. In many cases, we don't know the molecular, cellular, or even physiological mechanism behind these associations. And in even more cases, the therapeutic hypothesis does not obviously follow from the association. We don't know the target, we don't know the mechanism effect or the directionality relationship. So all this work needs to be done. Why is it so hard? Well, it's so hard because there's a lot of loci, but also there are research needed in many areas. And I love this figure because it kind of gives you the sense of the complexity of what it takes to go to mechanism. Also, why it's hard is because there are a lot of research needed, but also there is tremendous data generation in each domain that is needed to be layered and leveraged in different ways such that it actually gives you some sort of mechanism. And there are success stories, but they're minimal. This one you might be familiar with, being that you all work in diabetes, I presume. This is actually came out just this year from colleagues at ours related to the Lathe group, where this was for a zinc transporter. But the story behind the gene was SLC30A8. This was uncovered in, I think, 2007 from a genome-wide association study, one of the first genome-wide association studies for type 2 diabetes. They uncovered a common variant associated with risk for type 2 diabetes. But you fast forward to 2020, where you have a mechanism action finally published for insulin secretion of a loss of function protective variant that is also in the gene. So what this took was not only these first GWAS studies, but also deep sequencing studies that actually uncovered more variation in the gene, then also layering of model systems, expression analysis, annotations, and genotype callback to finally get you at a story associated with a particular variant. So this is wonderful, but we have to do this for hundreds of loci. So we have a lot of work to do. So for us, we thought about simply addressing a gap. And we tried to address this gap for all of diabetes. We thought the simple idea of making genetic and related genomic data broadly accessible and useful could hopefully have a significant impact on our ability to understand or treat human disease. And this by meaning we get this data in the hands of people who can use it. And so for that end, we were very lucky to work with the NIDDK and 5 industry partners to embark on a project that's now in year 5 called the Accelerated Medicines Partnership for Type 2 Diabetes, who as its central goal wanted to build a knowledge portal for type 2 diabetes to make this data more accessible and useful, and ideally support drug discovery downstream. And what I love about this collaboration, among many things, is that it's not just a scientific and academic collaboration in the sense that we're generating data, developing new tools, new analytical approaches, but also we're doing joint software development in an international space. So we have sites in the UK and in several sites in the United States developing software tools to bring this together into what you see in the portal. It's a quite exciting activity. So first, then as I pivot, I'm going to talk a little bit about what is needed to build such a portal, because this will help give us a sense of when you're looking at the actual modules, what is the underlying data and methods that come to bear. So first, there's 5 components. Let me talk about a little bit of them. First is the community. For this, representing the definitive and authoritative data from the human genetics community was paramount to us. We're very lucky. We work with this amazing community of people. But this really started as a grassroots effort. When the portal first began, there were about 9 data sets we were dealing with. We were dealing with a very manual and handshake process with our collaborators and colleagues, and us at the Broad to bring in individual level raw genotype and sequence data and deeply rich curated traits for diabetes and related complicated traits. And for that, we just want to store them, find a means to process them and harmonize them in a systematic way, and then represent them back to somebody that could use them. So this was done in a very manual approach for a long time. But what it allowed us to do was to design a robust and reproducible approach that is the authoritative representation of what the community underlining really wants represented. And that's really important to us. But to do this at scale, you have to build this into a software platform. So we've also, over the past 5 years, we built this into the platform where we can take in individual level data, process it with the best standards by the research community, and represent them in the portal. But moreover, if there are association statistics generated elsewhere, we also bring those in and can rapidly put those in the portal. But everything is documented such that you can always dig back to the provenance of the data. And this was done specifically in order to build this scaled infrastructure so we could do this in a rapid fashion. The one thing that's critical, I think the way we thought this is, Noah built the ark before it rained. And you can imagine that just having one site in the United States that can collect all the data in the world is limiting, because there are 2 challenges. One is there are geographic reasons by which data cannot leave a certain domain for ethical concerns or just because of the way the consents were written. So we're very lucky to work with the European Bioinformatics Institute to build a tactical replicate, if you will, of the analytical platform we have at the DCC at the Broad in Boston to build it at EBI. And it is a mirrored system such that individual level data can come in to that warehouse, be processed and analyzed in the exact same fashion, but represented in the portal in a way that as a user, it is seamless to you. Moreover, federation is critical because not everyone is disease. We are not domain experts in every data type, for example. So what you want to do is you want to capitalize on other sites who know how to process, store, and represent other data types better than you do. We're very lucky because we have a colleague funded under the AMP at the University of California in San Diego, Kyle Galton, who has adapted the ENCODE system to represent and store the annotations, genomic annotations, ChIP-seq data, HiSeq data. And these are stored at DGA, but then represented to you in a portal that is seamless to you as the user. And we're going to talk a lot more about those data later in the demo and how those data come to bear. Then once you have that foundational data, both the genotype, the human genetics data, and these annotations, there are many methods that take those two input data sets and run on them and produce more results. So you really want to take advantage of the fact that there are methods to derive pathways from these data sets, to run EQTLs, to look at rare variants, to produce a bottom line p-value if multiple cohorts have genotyped the exact same trait. So for that, we built what we would call a knowledge graph, which is a means to store and represent the relationships between the different data types we have stored at the portal and then represented for you. So these components all build our software platform. And this platform is expressed through visualizations that I'm about to show you with the portal. This is kind of all how it fits together. So with that, I'll turn to the portal. So the Knowledge Portal, just a brief history, was launched in February 2015 to about 70 original collaborators. It had 9 data sets, as I mentioned. And today, now, all you need is a Google-based login to log in. The reason we do that is so that we have a small terms of agreement just about citations and how you use the data, and also helps us track usage of the site and how many people are using it. So most institutions have a Google-based account these days, or you can just get your own. So once you have that, you're in. So getting familiar with the resource. When you log into the source, this is the first page that you see. This is the top of the home page. And if you want information on the AMP partnership, you can go right here and it will bring you there. You know, basic navigation at the top. I'll talk a little bit about where these resources are in a moment. But here's the guts. Right here, there's sort of 2 methods of entering the portal. One is to explore by a gene, a variant, or a region. I'm going to walk you through that. The other is to explore by phenotype. So we're going to go through those over the course of the meeting. So first is what types of questions you can answer. So let's start with something simple. First, at the bottom of the page, I forgot, there's all this information about the project. We have a blog post if you want to find out what's happening. We do those monthly, information about the project, and also links to our other knowledge portals, which I'll talk about momentarily, for cardio-metabolic diseases. So starting off. So what does the Type 2 Diabetes Knowledge Portal offer? The first, I talked to you a lot about the type of data that we bring in. So first, before you even navigate through different questions or genes, you might want to know what type of data are on the portal. So for that, you just go up the top here to our data sets page, and it will bring you to basically what we can actually say is the largest collection of definitive type 2 diabetes genetics data in the world. And they come in 5 different classes. Type 2 diabetes genetics community-derived data sets, publicly available GWAS data sets where we actually work directly with the collaborator to curate and represent the results definitively from that resource, and funded data sets to generate new novel data, and disease agnostic resources, which include like 1,000 genomes, and Nomad is also represented in the portal, and international biobanks, all totaling about 84 data sets for 191 traits. So how can you navigate them? So first, this is what the data page looks like. It has the data filters by GWAS data set, whole genome sequence data, exome sequence data, and exome chip. And you can browse by phenotype class. And so these are each of the phenotype classes. And the list will populate based on the data sets in that class. And they populate based on something that's new. New is in the last 2 months, and other data sets are listed below. And if you want to know for a given trait class, how many other traits do we have? So for example, glycemic traits, you can actually get dropped down and we'll filter by all the other related quantitative traits and specific traits we have curated for glycemic traits within the portal. And the data list updates dynamically to whatever you've selected. So that's for like all the data that's in this portal. But what we really feel is important for all these cohorts, we want to make sure we document the provenance. So we have comprehensive documentation and cohort description for every single data set we represent. What's critical to this is that it allows you to, if you're seeing a data set in the portal, you can always track back to the citation. You can often get download statistics right here. You also get information about the study design, the cohort information, the trait curation and preparation, how it was actually analyzed. And if we've analyzed it directly ourselves, you get extensive QC reports and analysis reports. This allows you to always track back to the results that you might see rolled up to you in the portal. So that kind of gives you a sense of what the data are, the foundational data. So now what questions can you ask of the data? So let's begin by going through it with a gene. So many of the ways you approach this is, I have a GWAS locus, so I have a region, so I would type in a chromosome position, or I have my favorite gene, or I read a paper and there's a particular variant that I'm interested in. So we're gonna walk through each of those real quick. So first, the gene. So this is SLC38, which I referenced earlier. This is, if you type in this gene, it'll bring you to what's called our gene page. And the goal of this page is to summarize the evidence for that particular gene across all the phenotypes in the portal and give you sort of a first blush of phenotypes with significant signals, so that's genome-wide, are in green, and in yellow is anything that is nominally significant. But this allows you to update this table based on any trait you're particularly interested in. So if you click on any of these, what I'm about to show you will update according to the trait you selected. It defaults to the first trait, and typically for diabetes, will default to diabetes as your first trait if it's diabetes that it is associated with. And it gives you a curated summary of the gene and some basic information. If you want to learn about the gene page, you can go up here and there's some guides. So then for the bottom of the page, there's 4 tabs which we'll navigate through. One is first the top variants. This assembles for you the common variation associated with that particular gene in a way that is by p-value, the strongest p-value, and biggest data set. This is all manipulable, downloadable. You can search through variants, and you can basically play with this table as you would will. But this allows you to sort of see a distilled view of how that genome-wide significant association was arrived at from all the genetic definitive data. Then what if you want to know about not just the common variants, but the potentially the coding variants that might impact function? So the deleterious possibly variants. You can go to the high impact tab, which is the tab right next door. Oops, sorry. And this will provide you the same table, but listed for the protein truncating mutations or the coding proteins. And again, these are organized in the exact same fashion for you, and you can click and navigate through this table as well. Below here, we've actually also computed for you aggregate variant tests for all the different masks associated with how you might collect the non-synonymous and protein truncating variants. So this gives you a view distilled of all the coding variation that may be impacting the association you're seeing in a particular gene. So again, this is distillation of the genetic evidence in one table for you. Then if you're more interested in navigating a region or looking around that gene, at the bottom of the page, you have what we sort of feel as sort of the classic way of viewing a region around some association statistics. This is locus zoom. This is done in partnership with the University of Michigan, and this is a classic viewer for viewing genomic associations in a region-based. So you get all the SNPs, the recombination hotspots, and the p-values associated with it. And you can click and choose what you want to do for tissues, data sets, or phenotypes, and you'll see all the genes down below. And this is also quite nice because you have the layers of the chromatin states for the relevant tissues below. So this gives you sort of a region-based view of your associations for that particular gene. Now, if you want to see things in a little bit more detail, you can manipulate this table. This is all on the fly, allows you to manipulate, and you can actually point and learn information about the different SNPs and variants in here, and the table will update associated with whatever variant you're choosing. And I like this because if you're interested in looking at a particular variant and its relationship to other variants in the region, this is a very valuable tool for that. But what about if you want to say, you know what, for this gene, I want to know what are the best variants I might want to study for functional follow-up. So this is your 4th tab. This is called the credible sets. And this is done from data that we've derived from our community. And this represents for this given locus, and this given region, and the genes around it, with SLC38 as the anchored one, what are the credible sets? What are the most likely variants contributing to the signal as predicted by the genetic evidence, but also the tissue annotation? So what tissues are they active in? What is it affecting in terms of weak enhancers versus active enhancers? And that information is all layered for you down here. And so you can navigate through this as well to know what is the best variant at that given locus. And if you were to see this, what happens in the credible sets tab is it automatically updates the locus zoom plot below for those particular variants and allows you to see the chromatin states and again the tissues. Oops. All right, so that's the gene-based view. So what about if you have a particular variant you're interested in? So this is, again, you're navigating through the genetic data, but you want to do it from a variant perspective. So this is very useful if you're reading a paper and there's a non-coding variant associated with a particular trait. So for that, you'll get our SNP-based summary, variant summary. So this gives you a compendium of options that you can view for a variant of interest. It says what are the different transcript changes, what are the effects of that particular protein change. But then you can click through this accordion and see a set of different tools to navigate through this variant. The first is the best, in my opinion, which is this variant at a glance, which is this top one. This tells you from the portal what this variant is associated with, all the traits in the portal from what we call the bottom line analysis. And I love this feature because what it does is, in many cases, we have data sets where there are many traits also run for those association, but oftentimes cohorts use the same samples, whether it's a cardiovascular cohort of diabetes, they may use the same samples, but they have information on more phenotypes. But what's nice is when you combine them, you actually want to make sure that you're representing the right p-value. You don't want to overrepresent it if the same sample has been genotyped. So what this module allows you to do, it calculates the bottom line p-value, but also taking into effect any sample overlap and removing that to give you the best results. So this is a computational approach that is done on the, in the software platform that I referenced earlier, but represented back to you. And that's a novel way of representing. And many sites just give you all the different lists and you have to figure out how to combine them. This does this for you. Another thing I like about this is you want to compare this variant if you want to know about the bottom line for the diabetes association, but it also says for the UK Biobank analysis that's been recently published that has 1,400 traits, we worked with our collaborators to represent the results for those in the portal integrated here. So it says for all these different types of trait groups down here, what other associations are for this particular variant. So you can kind of learn the GWAS, if you will, for this particular variant across what we have for phenotypes in the portal. Looking down below, if you really want to focus on just the portal-based data, you can look at all the different cohorts that we have in the portal and the associations relevant. And this gives you the direction of effects. You can filter this based on anything that you might like to see or particular call out. And it's filtered by genome-wide or locus-wide significance. And you can see the things that sort of are jumping out to you. And then if you want a deeper table of that particular graph, this gives you every single trait in the portal, the data set, the effect, the direction, and how significant it is. This is all exportable. This allows you to really deep dive into the different phenotypes within the portal. So that gives you sort of a sense of how you might look at the genotype, the distilled genetic data based on region, gene, or variant. Well, what about if you want to know about your favorite disease or trait? So this brings you to the phenotype page. So for this, we have several views, one of which I'll talk a little bit more detail a little later. So the first is you can type in any phenotype you want, and it will give you the phenotype page. This is how you navigate to it. And there's several things you'll get here. But first, you might just get all the genome-wide significant associations for that given trait. So this is what we call the interactive Manhattan plot. That's one feature you might see. What if you want to know what are the top gene-based results from my favorite trait? This also will allow you to see the types diabetes, best for the exomes, the largest data set we have for the exomes. What are the top gene-based results? You can have this for any phenotype for which we have whole exome sequencing data in the portal. But what I think is a unique feature about this is those are just results and summary statistics. What if you have your own question you want to ask of sequence data, for example? What if you want to perform custom analysis where you want to potentially, look in type 2 diabetics for people who have a high BMI, or slice and dice the database on a set of phenotype filters or SNP filters that you want to run? Well, we built a mechanism to allow you to interact with our exome sequencing data on the portal, but only put, and then it would run a custom analysis that you're asking it to, that you're predefining based on a set of variants or filters based on phenotypes that we have in the portal that you would run on the fly. And what happens is it runs on the data, the raw data, and represents you back to your results. This allows you to do a custom analysis that is not already on the portal and almost allows you to generate your own hypothesis. This is a very novel approach. We love this because it doesn't ever expose individual results, but allows you to have individual access to data. And this is what it gives you for the results. So that's another key feature of the portal. And this can be found in our analysis modules page. You just want to directly dive into these different modules. It's also integrated in the gene and variant pages, but there also are other analysis modules which I don't have time to go through today. So finally, that sort of wraps up the current features of distilled representation of genetic data for non-experts. Where are we headed next? So I feel, we've done a good job of representing the human genetics data from just the distilled, what are the p-values, what are the associations, the direction of sex, how do we bring this definitive data to a way that a non-expert can interact with it. But the next step harkens back to what I mentioned earlier. There's research needed in many areas. How do we bring in all these other data types and leverage them to help inform these 403 loci that we've identified for type 2 diabetes? So for this requires a shift in focus. This allows us to focus on trying to figure out how to allow people to determine what is the variant, what is the regulatory effect, and in what tissue, what cell type. And some of the things we've done already, but doing a better job by bringing in other data types, as I list below, what is the pathway, what is the mechanism? So to that end, what have we done so far in that space? So one of the nice features that recently came on the portal was something that I'll show you now. And this gives you an insight as what is known about a given disease. So for type 2 diabetes, we have a really wonderful feature, which is our first expression of what is known today for predicted type 2 diabetes effector genes. So what this is a representation of layering of evidence, coding evidence, regulatory evidence, and perturbational evidence. It's really an application of a heuristic developed by Anuba Mahajan and Mark McCarthy, who are colleagues of ours, that we've now represented in the portal. And what you are able to do here is for these different classes, which you see here, you can see, and actually this gives you a distilled view of all the genes in there, just to know that this is actually a application of someone's heuristic that we embedded into the portal and now represent to you. But what you get is a complete list of each gene and its supporting evidence. So you have the different prediction level that it is, the actual gene, which you can click the gene page and then drill down into the deep learning. But you have the 3 levels of evidence, the genetic evidence, the regulatory evidence, and the perturbational evidence. And for each of these, you know how we determined it, how is it defined, and for each of them, moreover, you can then expand the table and you can click and actually get to the evidence supporting that particular reason for why we made that particular gene a strong candidate for being an effector gene for type 2 diabetes. And what this is very useful is you can also then this also gives you the mouse and then the regulatory and perturbational evidence. So it gives you a way to both say, what are the genes in this list and what are the different levels of significance, but what is the supporting data? Where did it come from? What's the rationale? And we really hope that this is actually something we'll be adding more to, because we want to take from these 403 loci and actually really make strong cases for what are the genes and what are the variants, and really build on this whole model for diabetes and moreover in other traits. So to do that, we need to address these new gaps. We need to understand what other new data sets we need to add to this list to do this for other phenotypes. What is the information that must be captured and retained and thereby represented back to you so that we actually can make inferences as what is the variant, what tissue is it active in? And then how do you represent these in a way that is useful to them? So we've got some inroads, and I'm going to show you right now. And what can you do in the portal today? So first, our goal as we're doing this is to catalog all these resources so that you can go from the causal variant to the disease, because within there are so many different orthogonal data types and representations that you need to bring to bear. We're very lucky because, in human genetics, we've had a lot of foundational and computational advancements in the last few years that have allowed us to have these foundational data sets available to us, but they're not in one place and they're not integrated, and they're not, moreover, represented in a way that you can make use of them. So our first blush at that is to take a set of example data sets and data types, and bring those into the portal. So I'm just going to walk you through a couple examples of what those data set types are, and then we'll show you how they're expressed in the portal. So one example data set is a TAC-seq. This helps us understand chromosome accessibility and helps with predicting of variants with regulatory effects. So this is very good for non-coding variants that have been uncovered. That's one data type. Another one is promoter capture C. This allows us to predict the variant to gene links. So this is also useful for non-coding annotations. This is another data type that is a new data set that we need to help layer into the genetic information that we're presenting. A third example is a method, or EQTL. This gives you clues to function. It predicts variants that affect both the trait and an expression of a gene. And that is very valuable because then you know what tissue is active in, and it helps you point to the particular variant. So for that, this brings us back to that knowledge graph I referenced earlier. This knowledge graph allows us to layer this omics data in a systematic means to represent it on the portal. So what can you do today? So this is a great example. This is the K1D locus, which has been implicated in type 2 diabetes. So to get access to this information, you go to the same gene page, but you go to this tab over here on the right called Genes and Region. And actually, first, before we do that, let's first go at the bottom of the page where you can see from the locus zoom plot, if you want to get some of these data sets represented to you, you can click on the locus zoom plot, click on your tissue. Here I'm clicking on pancreatic islets, and you will see the attack-seq results for chromosome accessibility for that particular tissue. So where are we getting this data? Well, you heard me referenced earlier that the DGA is a place to store this, the Diabetes Epigenome Atlas, our federated node. So if you really wanted to drill down to what that represents, so what we represent for you was a summary statistic for the chromosome, the peaks of the attack-seq. If you actually want access to the results that underlie those, you can go to our sister resource, which I pictured here. You can search for that exact data set, and you can see it right here with the 37 data sets and the islets, and then you can access it directly if you were particularly interested in it. So as we grow this resource, we want to make this more integrated. But right now you have two means. One, to see it in a distilled view on the portal as part of the genetic data. But then if you're particularly interested in that data type to actually have access to or run computational methods, you can use the DGA as a complementary approach to have access to that data as well. And so what's nice is we are building that infrastructure such that, we at the DCC will be the house with the EBI for the human genetics data, but the DGA will allow us to represent and store all these orthogonal genomic data sets. And then over the next couple of years, we will be representing ways to represent those in visualizations and different tools that make them useful. And so one tool that has made them a little more useful is this Genes and Region tab. So for this, what we try to do here is allow you as a user, if there's a locus you're interested in, you can type in that locus and you'll see for the genes implicated in that locus, what are the potential results for computational methods that may be useful in helping to determine which gene is the most likely gene at that locus to follow up for functional study. And by doing this, what we're doing simply right now is just allowing you to see different lines of evidence. We allow you to see the genetic data at the top, which you can see elsewhere in the portal, but we also bring in things like predictions for tissue predictions with meta-XScan. We allow you to see pathway-based with predictions and prioritization by gene, prioritization by depict or gene set enrichments. And also there are EQTL methods represented here. And then finally, there also are model systems. So this is our first blush at getting you results run across all of the data in the portal represented for you that a way you can navigate. So each of these is clickable and you can see the top hits and you could start to have a sense for yourself what results are useful for you to make predictions of which variant to study. Now, this is clearly the beginning of it, but it's the first place where we're starting to integrate these orthogonal lines of evidence and represent them back to you. So what we're hoping is that ultimately we'll be at a place where it's not only distilled data represented for human genetics data, but also genomic data for non-experts. So over the coming years, that's where we plan to move with the evolution of the portal. Now, one of the things we've heard from many of our users, and many of our users are computationally savvy. So what we've done just recently is to take from all of the underlying software platforms and different resources, one way is accessing through the portal. But if you're computational savvy, you would really like programmatic access to these results. So what we've done is enabled APIs for all of our portal results. And these are accessible through the API button right in the data tab. And if you are interested, every single set of results on the portal can be accessed through an API. So this is the user interface. This documents all the APIs and what they actually produce for you. And if you click on them, you can go to Swagger, which houses all of the different commands for each GET call for every portal module. So what this enables us to do is people who are interested in running their own methods or running a query that you would have to run over and over and over again on the portal to get a set of results, you could actually do it in batch. And this allows you to really, make our results accessible not only to a non-wet lab biologist, for example, who's not computationally savvy through the portal, but also someone who has a method or approach they want to run from a computational perspective, they can do it at scale. So that's been a recent development for us. To kind of just wrap up, I see these as kind of the marquee questions you can ask with the portal right now that we do really well. So what are the genomic associations for type 2 diabetes? from the and related traits from the definitive data from the community. What are the critical sets associated? What were the best variants for me to study? What are the most up-to-date and curated lists of predicted type diabetes effector genes, along with the supporting lines of evidence? And then what are the results of computational approaches and relevant genomic annotations to assist me in the prioritization of a variant or gene from a locus? So that's kind of where we are. Just to kind of give you a sense of who's using the portal, I think this is kind of interesting. It doesn't have a huge user base. It's had 20,000 visitors since its inception, but we have about 15,000 registered users. And it's averaging about 100 people a day. But the nice thing is they stay. The average use is about 7 to 8 minutes, which means they're not just looking at the front page and leaving. They're actually trying to do some of the different features and modules. We've had over 80 citations and 17 to date synchronized data releases with publication. This has turned out to be a really valuable piece of the portal. I'll talk about that in just a second. We're definitely, our users are driven by data releases and public events. We have a lot of public events. That's where our usage goes up. Also big data releases. This has just been interesting for us to learn that that's really the most important tool that we have to really get to our users and connect with them. What's lovely is that the ethos I think that we've developed for the Type Diabetes Knowledge Portal was contagious. I think the field as well in complex trait is struggling with the exact same challenges. There's collaborative alignment. People want to make the barrier for sharing data lower. They want to have a means to share their definitive results, knowing that it's coming from those communities, but also making them accessible to anybody. So we're very lucky with the software platform. It's readily adaptable to other diseases. So what we've been able to do over the past couple of years is adapt this platform to other diseases. We've started with diseases that we know really well and that we believe are valuable to type diabetes research, which are other portals in the cardio-metabolic space. So there are 3 other publicly available portals for related traits to type diabetes, one being the cerebral vascular disease portal that was launched in 2017. It's open access. You can see information about it here. It's got about 4,000 users. It's got the largest definitive collection of stroke genetics results. What's lovely, it's got every single hand-curated stroke phenotype, of which there are about 67 of those specific traits just for different stroke phenotypes. And that you wouldn't get on another resource. Same for the cardiovascular disease knowledge portal. This was launched in 2017. Again, the definitive source for cardiovascular genetics data. This was done in partnership with folks at the Broad, St. Catherine and Patrick Eleanor. And most recently, a sleep disorder knowledge portal was launched in 2018 for sleep disorders, again, being the definitive resource and open access. So what's lovely about this is they're built on the exact same platform. They have the exact same tools, but they have the definitive data from that particular community. And that's what's been really a lovely thing for us to work with them. You can get access to these on the homepage. They're all connected. But what we've really felt is that to garner the best value from these independent portals built on this, particularly if they were related, is to bring them together into one resource. And we're very happy to announce that coming in 2020, we will produce a common resource across metabolic diseases where there'll be a new interface that allow you to navigate through all of the traits and all of the data sets currently in these 4 portals easily. But also, if you're interested in just looking at the cardiovascular data, you'll still be able to navigate to that particular site. This is important because we want the communities that generated and represented their definitive data to always have a place where they're represented. But we want to make it so that a user can navigate across lipid traits all the way to glycemic traits in the same resource. So that'll be coming in the next year. It's, again, the same platform, the same architecture. And we also have some other resources where we've branched out into non-cardiometabolic diseases, and this is accessible through our knowledge portal network page that describes all the documentation, the API access, our GitHub repos are here, and also another portal for ALS, which is in a very different disease space, but you can check it out if you're interested. If you want to learn about the portal, there's live and online learning. We have videos, tutorials, bi-monthly webinars, which you're welcome to attend to. Those are specific. We usually do a feature, or we feature a scientist talking about a method or tool or data set and how that's going to be represented in the portal. So we can invite you to those. But these are also available to you on the portal if you want to learn about any of the features. This team that we work with is amazing, a team of software engineers and computational biologists and managers who have been wonderful to work with to actually build this resource. We're also very lucky to work internationally with a set of people, as I mentioned before, from Michigan, Oxford, and UCSD to help bring this joint software development system to you. And then finally, the AMP partnership, which for the past 5 years has been remarkably supportive in us doing this activity and giving us great feedback to build the resource you saw today. With that, I'll take questions. I hope I left us some time. Oh, it looks like I did, but thank you.
Okay, now you can unmute yourself and ask question. Let me see.
Oh, hi, this is Marian. Thank you so much, Noël, for that. Unfortunately, I was a little late joining, so you may have answered this question already. But you mentioned definitive data sets quite a few times, and do you have criteria for what are considered definitive data? You're muted, I'm sorry.
Our criteria for that is the community or the people who derive the data have said that this is their results. So in the sense that they are sending them to us and they're sort of stamping them as their authoritative results, whether they're published or they are saying that these are the ones that they want represented. So that's how we define it as definitive. In the sense that we're not reprocessing it or doing it a different way. We are using their approach, their methods, their trait preparation, and representing it to the portal. So it's from them. That's how we see it as definitive.
I also think there's another sense that the community responsible for developing the knowledge portal consists of roughly 100 or so T2D investigators, including many of the leading experts on GWAS and the state-of-the-art methods for interpreting those associations. So there's a community agreement that these data sets are the ones that they trust and use in their research. So that's also the component. So a lot of it is omitting some of the data sets that may be small or may be outdated or may not be what the community anymore considers to be the standard. So a lot of it is with having the experts who actually use the data being involved in deciding what goes in the portal.
And are you seeing, as more data sets come in, at some point do you kind of saturate on what the results are, in the sense that they do arise to consensus, or are things changing a lot, you know, depending on the data sets you bring in? I don't know if I phrased that correctly, but.
Yeah, I guess, by results, do you mean like the associations, or do you mean?
Yeah, you were showing associations across data sets, and one wonders how many do we need, right? And when do you start seeing like a fluctuation coming in? Like if you have 2 data sets, are you sure? But then by the 3rd data set, it becomes unclear.
Yeah,
I think that would be.
So I mean, that's actually a pretty deep question. As we get more and more data sets, more and more loci become significant, which is expected because currently only about 20% of the genetic basis of diabetes is explained by the associations in the current data sets. So any new ones that you bring in will add more. Now, Noël described the bottom line analysis, where we actually do additional analytics that, rather than trying to simply say if a new study observes an association, is that real, or should we add that, or is it invalidated by a larger study already in the portal? What we found from that is that actually sometimes new data sets will actually remove associations that used to be significant in smaller data sets.
Right.
So to that extent, that improves. Now, the third question, which kind of gets at your how many do we need? If your question is, are we learning new biology by bringing in more and more associations? That is a question about which there's a lot of disagreement in the field, and which is responsible for the increasing pivot towards augmenting the GWAS data sets with a lot of this functional data that can help get at the genes and pathways that are underlying them. And that's an ongoing work that we're doing.
That's interesting, because it does become a question of how much of the data do you need, and when is there flux? And it really, I'm not a GWAS, but my relatives are all in ancestry.com, and every week they're like, oh, now I'm 20% Swedish, and I used to be only, I used to be no Swedish, and I'm like, stop listening to them.
Right. Yeah, I mean, the core, so the answer is like kind of the core state of the field has kind of been static, right? So there, for instance, there was a 2 years ago, like a 900,000 sample GWAS published by our community, and it had like 400 associations. And now there's a larger one that is on bioRxiv that has 400 more or something. And people, I think, have for some time no longer really been excited by new loci anymore. They don't really add to our biological understanding at this point. So it's pretty recognized that the state of the field, which is the genes that we trust in are really being investigated, and the methods are kind of stable. And it's really at the fringes where you're starting to see like a bunch of new loci. So it's not like we're going back and revising 20% of the genes or things like that. It's pretty stable what we are really focused on at this point.
Well, it's good to hear.
Yes.
But thank you. It's a really interesting portal, and look forward to poking around in it some more.
Thank you.
Just one question from the chat room, probably easier. So is I think he's asking if a research want to contribute data to type 2 diabetes knowledge portal, do they need to be a specific member from a consortium? Or you mentioned there are 5 type of data sets. Do they need to deposit their data to those database?
Great question. They don't have to be a member. In fact, we really want to make this a place where any data set that is relevant to the portal can be represented. And so for us, we want to partner with anybody who like their summary statistics or whatever data type they have. I think the place to start is with us, the Data Coordinating Center, and then we can decide whether the data set needs to be loaded through the EBI or through the DGA. It all depends on the data set, but we can start with us. So in fact, on the portal, there's a data submission website, a link where you can actually email us directly, and it comes to me and a couple other people if you're interested in submitting data, and then we follow up with you. And for many data sets, it's a very easy and streamlined way to upload and bring in your data. And some of them we're just learning, as I mentioned earlier, with some of the more functional data sets.
Are there any more questions?
Yeah, this is Marianne on that issue. I've been hanging around a lot in data repository world because, there's an effort to really upgrade and modernize the system of data repositories. And in your portal, it looked like most people had their data deposited someplace else, and then you were taking it over. Do you, when somebody contributes data to you, are you taking primary stewardship of their data, or are you putting it in EBI or NCBI or some other place to manage the data, and then you take a copy and add it? How is that working?
So typically for many of the 4 public portals that I mentioned earlier, we are the place. So they're actually setting it in the place, more increasingly so. Particularly for the GWAS data sets and sequence data sets, because it's pretty straightforward and simple, and it's really nice because it allows early access to summary level statistics. No raw data, right? That goes into dbGaP or whatever.
Okay, that was my question. So you're not taking the raw data, that goes someplace else. That's what I thought.
No. We do take the raw data if they're interested. So for many of the diabetes activities, we have the raw data for all the exomes. For all the exomes on any of the portals, we have the raw data that we've represented. But they're also deposited in dbGaP in many cases, just by sort of requirements for funding, and also just to make sure that there's many means for people to get access to the data if they want to do things differently. So like the BAMs can come from dbGaP, but on our portal you can actually run custom analysis, for example.
Okay, thank you.
There is one more question in the chat room. So can you explain in detail about developing new hypotheses? Does this mean that we can explore different phenotype to genes that are predicted to be associated with already in other phenotypes?
Yes.
Good question. Yes.
So in terms of in detail how to, so is the question about generating new hypotheses or testing existing hypotheses?
The question comes from Sampath. Do you want to put more?
Okay, both. So I'd say there's 2 primary ways that you could envision generating new hypotheses. The first would be if you have a set of phenotypes that you are interested in, that you believe are relevant to some pathway or some condition. So say you're interested in a pathway that is like insulin resistance, and so there's signatures of certain cholesterol and waist tip ratio and diabetes levels. You can identify genes that might be relevant to that pathway by using the variant finder that Noël demonstrated, where you can construct queries of criteria for each trait and even direction of effect, and then identify genes that fit that. So an example of this would be, there was a paper published that there's an insulin resistance signature that's like higher fasting insulin, higher T2D risk, higher waist tip ratio, lower triglycerides. You could do a search on the portal to find every variant that has an association with that pattern. So that's one way, and that was designed in large part because drug companies often think this way when they're looking for targets that might have a desired profile. The second way to generate a hypothesis would be if you had a gene or you had a variant that was associated with the phenotype, and then you wanted to potentially hypothesize its mechanism based on the collection of associations that you observe for it. And you would do that by either looking at the gene on the gene page, and Noël showed you how to do that, and seeing what's associated with it, or on the variant page, typing that in, and you see the full pattern of association. So I think those are the two primary ways I would envision generating new hypotheses. There's some more nuanced ways around causal variance and whatnot that are probably a little bit more nuanced. I think for testing hypotheses, it's something that, to be honest, we don't really support at this point. You can certainly test if you say, is my gene associated with something? But the problem is we don't test the negative of a hypothesis. We can't tell you whether the hypothesis is rejected by the data simply because GWAS isn't really set up like that. For example, if you do not observe an association for your gene, that does not mean that the gene is not relevant to that trait. So I would say at this stage, the portal is mainly geared around generating hypotheses and gathering data that you could then officially test through follow-up experiments. And in terms of actually providing guidance on what that implies for testing ones is something that we're currently thinking about, but don't have perfect solutions for yet.
Cool.
Okay. Francis,
Great, thanks.
for clarify. Yeah.
Cool.
Right. So like the issue with the negative hypothesis is just that if I don't observe an association for my gene, one reason could be that there is no variant that perturbs that gene. And that's kind of, so the caveats would be like, if there is so one place where I guess you could test it, this is maybe if you have a variant and you want to know whether the variant, if it's associated with one trait, is associated with another, that is something that the negative result can probably give you some insight that it doesn't appear to be there's association. If it's, in other words, if it's the same variant. Although then there's issues still with like, do you have enough power in your data set? And those are things that we could surface, but don't do so at this point. So I would not, so anything that, if you observe not an association, I would not immediately conclude that your hypothesis is rejected. But it is something that, we could potentially, like one of the things we often encourage people who are doing this sort of thing is to contact us because we can help by taking a look at our data and giving you guidance on whether the hypothesis is really rejected, if that's important.
Okay. Yeah, let me check. So are there any more questions? So if you have any more questions, please feel free to send us an email. I can forward it to Noël or Dr. Flannick. And thanks for Noël and Jason for great presentation and then spending time with us. Thanks for everyone joining today's webinar. I hope you enjoy today's webinar and have learned a lot. I personally learned a lot. So a recorded version of this webinar will be available on dkNET YouTube channel next week. We'd like to encourage you to share today's webinar with your colleagues and students. Our next webinar will be on Friday, March 13th, and we will introduce you another NIH-funded diabetes resource, the Human Islet Research Network. To receive upcoming webinar information, please subscribe to our newsletter or check announcement at dkNET.org. Thank you again and have a great weekend.
Thank you. Take
Thank
care.
you.
Thank you.